Let's start. Hello, everyone. Thank you for joining us today on our information webinar about our recently opened call to address prolonged retention of oral peptide formulations in the gut, sponsored by Novo Nordisk. My name is Arash, and I'm a scientific recruitment specialist at BioMed X, and I will be moderating today's webinar. Today we have John Lindner and Thomas Huckle from BioMed X, and Kelly Vendera, Stephen Buckley, and Marianne Lind from Novo Nordisk as our speakers. That being said, I'm gonna ask them to kindly introduce themselves and give a brief introduction about themselves. John, can you please start?
Good afternoon or good morning, everybody, depending on where you are. My name is John Linder. I've been with BioMed X since 2018, initially as a group leader and currently having a dual role as head of research for our United States-based projects, which is less relevant for this call. More relevant is my role as our global head of talent sourcing, which means I cover scientific aspects of our recruiting process from the opening of the call through to the last day of the boot camp that you'll hear about in this webinar. This project will then be passed over to the hands of Thomas in Heidelberg.
Yeah, thanks, John. Good day, everyone. My name is Thomas Huckle. I'm the head of research for European projects at BioMed X. I've been with the organization for around about 5 years. I'm overseeing all the projects in Heidelberg and other European countries. With this, I'm heading over to Novo Nordisk colleagues. Who wants to start?
I can start. Hi, my name is Kelly Vendera, and I'm a senior scientist in Novo Nordisk in the Department of the Oral Drug Delivery Technologies. I work in the peptide oral delivery area. For my background, I'm a pharmacist by training. I graduated from the National and Kapodistrian University in Greece. I did my graduate studies in pharmaceutical technology and pharmaceutical biophysics in the UK at King's College London, following a short period of 2 years as a teaching fellow at the same university, before joining Roche in Switzerland for a 2-year postdoctoral position in the area of oral delivery of oligonucleotides. Before joining Novo Nordisk. So overall, my research focus has been in the formulation area, from designing polymeric nanoparticles to polymeric coating of Lego type tablets, and then drug and cyclodextrin in liposome formulations for antibiotics. In the last 4 years, I'm focused on the oral delivery of large molecules. I'm very excited to be part of this webinar. I'm going to be also one of the coaches during the bootcamp in the industrial mentor.
Thank you very much. Stephen, please.
Yeah, very happy to be joining today. My name is Stephen. I'm Vice President Scientific within Drug Product Research in our research and development organization at Novo Nordisk. Been at Novo Nordisk for more than 10 years across different scientific and leadership roles. Have a background similar to Kelly as a pharmacist, and been working broadly across drug delivery and formulation sciences, both as it relates to peptides, proteins, and other modalities. Very much look forward to today's webinar. Marianne, I'll hand it over to you.
Thank you, Stephen. Hello, everyone. My name is Marianne Lind. I'm also from Novo Nordisk. I'm a senior principal scientist in the same part of the organization as Kelly is part of, where we focus on oral delivery technologists. I'm a pharmacist by training, like Stephen and Kelly. I have a PhD in oral drug delivery. Both degrees are from University of Copenhagen. I have recently joined Novo Nordisk a few months ago, but I have more than 15 years of industrial experience within drug delivery, various types of drug delivery, various administration routes. I have always had the focus of this special interplay between formulations and the excipients that we choose for formulations and the effect on the biological membranes, the oral GI tract in this case. So I'm also very much looking forward to this exciting collaboration with BioMed X, and also to receive your applications and meeting some of your applicants at the bootcamp, where I will also be a bootcamp coach. So I'm looking forward to that.
Thank you very much for your introductions. Before getting started, I would like to ask all of the attendees, if you have a question, please submit it in the Q&A box and not in the chat box. However, I have inserted 2 links in the chat box. One is for the call details and the other one a direct application link. I would like to mention that this call is being recorded and will be available after 24 hours and will be posted in our YouTube channel. That being said, I'm gonna start sharing the slides and hand it over to John to continue.
And I will just take that to, again, let everyone know we're very excited to have you here before I pass it back to Thomas to start going through explaining BioMed X as an organization.
Yeah, so shall I go ahead? Yeah, good. Yeah, I'm very excited to be here today. I forgot to mention earlier that actually I'm a chemist by training, so this is a project that is close to my heart. So yeah, this webinar, the coming hour, is about a new call for application. We want to give you guys all around the world some appetite as to what is behind that title. Can you go back? Sorry. Of prolonged retention of oral peptide formulations in the gut. Yes, next, please. So yeah, I want to introduce to you the BioMed X Institute that you know as an applicant, and if you win the bootcamp later, who is your employer. We call ourselves a special place where external innovation hub for pharma is residing. We started actually 12 years ago on the campus of the University of Heidelberg as a biomedical research institute. Nowadays, we are embedded in 2 campuses, one at the University of Heidelberg still, which is our mothership still, and on US ground, we are next to Yale University on the East Coast. And really, we are combining the best of both worlds, the academia on one side and industry components of the other side. I'm going to explain in a second what is behind those 2 pillars. And really, we over the years, we have refined a unique innovation process that defines us, describes us, and we published this in 2015 in Nature Biotechnology. So yeah, we see biomedical innovation by combining the best of both worlds. On one side, next click please, the industry, as you see here today, Novo Nordisk, our partner, with whom we define the challenges, from whom you get mentorship, from whom we as BioMed X get the financial support to conduct the research. And the other pillar on the other side is academia, you guys out there, the talents, the project ideas, the unique sparks, but also the mentorship, because all our projects have actually an academic, a professor at a university who gives insights, human samples, academic IP as well, the whole infrastructure that we can tap into on our campuses, be it here at Heidelberg or the Yale University. And combining those 2, we seed biomedical innovation and therefore provide solutions to our pharma partners, be it drug targets, be it biomarkers, be it AI models, or be it, for example, in this particular case, a platform technology that Novo Nordisk can actually use for their peptides to really make a game-changing event in oral drug delivery of peptides. So yeah, innovation as a service is our newest logo. We build from scratch what pharma needs next. How do we do that? We actually start discussing with our pharma partners in very focused sessions, identify top challenges, top ideas, top themes where we say, what would you like to see being a thing that moves the needle significantly the next 5 to 10 years? We identified an internal champion. In this case, it's Kelly and her colleagues. And then we have a call for application, which is well defined in this case here. And with over 8 weeks call, we do a crowdsourcing all around the globe, where we identify the 15 top talents, project ideas that together with Novo Nordisk, we review. Typically, we get 200 to 300 applications coming in. We thoroughly review them over hours and hours and select, as I said, the 15 top talents, who I invite to an ideation workshop, 5 days boot camp. In this particular case, it's going to be in Heidelberg, where the 15 individuals will be divided in 5 teams of 1 group leader candidate and always 2 postdoc candidates. And during those 5 days, under the mentorship and coach of the people and colleagues you've heard today, and John and myself, we will help you to come up with a unique project proposal that is presented at the end of day 5. And then the winning group leader has the opportunity to build a team and execute a 3 to 5 year project at BioMed X, in this particular case in Heidelberg, and build an innovative solution for Novo Nordisk. Next. So yeah, this is a photograph of the most recent bootcamp that we had with the pharmaceutical company in Japan, Daiichi Sankyo. A crowd of young scientists. You see John in the center. You see me somewhere in the back. We all wear t-shirts. We all laugh. We all have a great time during those 5 days. It's exhausting, but it's very inspiring. And it's a unique experience, even for those who go home and say, oh gosh, I have not won this bootcamp, but they all take away something that is unique. We have been, as I said earlier, around for 12 years. You see here an exhaustive list of all our partners who have trusted us so far in our innovation model. In Heidelberg, we have started 24 projects in various fields: oncology, immunology, neuroscience, platform technologies, and more recently also in artificial intelligence, to be contemporary. Next. Actually, yeah, we started in 2024 on the US ground. This is under John's responsibility on the campus of the Yale University. Was our first project that was sponsored by AbbVie. Around about the same time, we extended our model. We put actually BioMed X in a so-called virtual box and put it on the campus of a pharmaceutical company. In this particular case, it's Boehringer Ingelheim in the United States, in Ridgefield. We have 1 project there. We're soon starting a 2nd project there. We currently have a call for application. I think it's still open. Maybe it's still open. John, you may know better. We have 1 project here in the field of immunology. So this is BioMed X in a box. Then in the meantime, since 2021, we started 5 startup companies, slightly different model as to the Heidelberg and Yale approaches, or the X-seed approaches, where we have teams. Here we create small biotech companies, startup companies initially in the field of artificial intelligence. We did this in collaboration with Ion Labs in Israel, Rehovot, next to the Weizmann Institute, also prestigious place. And very recently, we were extending ourselves into the field of global health. Together with Gates Foundation, we have the call for around about 10 projects in the field of women's health. And our first project will start soon, so stay tuned here as well. This is another field where we are executing. Yeah, with this, what's in for you? What's in for the talents? It's a fully funded project for 3 to 5 years. You guys can focus on outstanding research and publish at the same time, so that after that time you still can decide, I want to go back to academia, or I got so much appetite for industry, I want to go to industry. You have the opportunity to cross-cultural teamwork atmosphere, because you may have seen on our website, we don't have 10 formulation teams. We have 10 teams of all different science backgrounds, of all different disciplines and therapeutic areas. You get guidance from experienced mentors, as you see here today. They come from academia, they come from industry. And it's an outstanding environment with direct access to leading research institutions and companies. Just simply in Heidelberg, there is around about 15,000 jobs just in research on the campus, and we are really in the middle of it. So we have framework agreements with all the major institutions there for special equipments, for special access, and so on and so forth. Yes, and you will get in addition leadership and entrepreneur training. if in case you want to decide building your own company, for example. And it's a risk-free assessment of a future career, either be in a startup, big company, or want to go back to academia. An ideal place, BioMed X, to jump into the next career step. Yeah, the hiring process, the winners of the bootcamp. It's going to be one group leader who's going to be the winner. And this group leader again has the opportunity to select 2 postdocs and 2 research assistants. And I'm spearheading already one of the questions that potentially would come from you folks today. These postdocs or research assistants could come from the bootcamp, but not necessarily. They could come from the team, but not necessarily. They could come from another team. But all get an employment contract offer from BioMed X Germany for up to 5 years. We will help you guys to relocate to Heidelberg, look for a work visa, finding a new home, moving a family, helping you guys as well, finding jobs for your spouses, kindergartens, and so on and so forth. The anticipated start date for this project is in spring next year, March, April. The bootcamp is going to be in December. I guess it's the next click, is it? No, it isn't. Yes, exactly. The bootcamp is in the second week of December. And 4 months down the road, then would be the potential start date of this project. Yeah, this is all I wanted to say about BioMed X, BioMed X in a nutshell. And I'm handing over to my colleague, John, who gives you a taster more from a philosophical aspect, who we are at BioMed X.
Thank you, Thomas. In the 7 years I've been with BioMed X, we've changed a lot. We've grown. And we've adapted. We also made it through the pandemic, which is all sort of a distant memory. But one thing that hasn't changed is our core values, what leads the way that we work. And in my opinion, these didn't need to change because they were excellent from the beginning. There are 3. The first is to cross leverage. This is sort of what I call scientific diversity. Everyone who works at BioMed X, whether you're in a team working on formulations for oral delivery of peptides, or whether you're developing sensor technologies for real-time monitoring of analytes in the blood or the brain, or if you're an immunologist, hardcore cellular immunologist, we all have protocols and we all have perspectives. And those fresh perspectives coming from an outside field actually enrich our environment. And we encourage everybody to talk with each other. There are no walls, there are physical walls, but there are no walls in between the teams except for the raw data in the lab notebooks and anything that is IP-able with respect to the sponsor. But beyond that, we all share our expertise and insights, and the tantamount of this is our weekly global institute seminars, which cover at the moment 2 geographies, soon to be 3, and next year 4. So these are fantastic virtual meetings where everyone in the organization gets a chance to present their progress at least once per year. The second core value is to cross-pollinate. For me, this is more about cultural diversity. We have experienced that with teams of 5 individuals, there's not a lot of room for clique formation. You can't have sub-teams that get along really well with each other and partner up against the other group members. And so what we do is bring in diverse backgrounds of individuals to Germany. Actually, German passports is probably the 4th or 5th most popular passport in our organization. So our working language is English, even though we are a German company. And all of this sort of forces individuals to coalesce together in a team. And we provide hiring support to the group leaders to make sure that their team fits the needs in order to function at a high level for 3 to 5 years during the project, especially when things aren't working. Science is what science will be, and there's always going to be that 3-month stretch of tough experimental results. The team needs to band together, and our solution to this is to embrace the diversity of the team composition as it actually creates innovative solutions to problems when everyone's working together. And finally, my favorite, seems very simple on the surface, but there's a lot under the surface, under the water, to aim high. We never want to be a Me Too organization. We're still searching for that corresponding author, Nature, Cell, Science publication. We believe that any of our new groups have an opportunity to get there, even though the bar is very high. We aim that high. in terms of publication. We aim high in terms of IP generation for our pharmaceutical partners. We want to contribute to the new generation of drugs that makes it to market, to the new generation of targets for some of our other teams that are doing target identification, to platform technologies that can accelerate the discovery of both targets and new pharmaceutical modalities. And just a note on aim high in 2025, for all of you, anybody can take the call text from our website and copy paste it into ChatGPT. We have done this. And if it worked, we wouldn't have a call for a team for 3 to 5 years in Heidelberg. So I ask you, please do not take this route to coming up with your unique, innovative, high-aiming proposal, because recent calls have shown us that many people take this route, and there are 6 evaluators on the project that have to read these proposals. So please respect their time as well, because what we see is 20 applications that all read more or less the same. They're all the same generic proposal to solve the problem. AI can't do it, guys. LLMs are not there yet. This is why we're looking to you for the creative solutions that we need. And with these core values, we are able to attract exactly those creative types of people. So on the next slide, you'll see where we've been able to attract talent from. And many of you may even see your own affiliation or institute. Here, we come from universities across the world. We come from industrial backgrounds. I myself did a postdoc at Novartis in Basel. They're in the middle of the screen. We've still got it up. And we really concentrate that talent at first in Heidelberg and now at our global sites. And while at BioMed X, we still collaborate with both those original institutions and new ones. So I think this number is actually now up over 70 academic collaborations with labs across the world that we've established, sometimes for sourcing materials, sometimes for production of various proteins or DNA, and sometimes leading to co-authorships in higher impact journals that we have been able to publish in. Finally, after their tenure at BioMed X, what is the employability of our fellows? It's actually quite good, and it's about 50-50 between what Thomas mentioned, going back to academia with the experience in the pocket, or moving on to industry. So we do offer all of our fellows sort of a free look, and we offer our pharma partners sort of an extended interview process, a lot of visibility for our fellows to obtain opportunities for themselves in either of these paths. So if we've been able to excite you so far, and if the colleagues from Novo Nordisk are able to whet your appetite for the project itself in a few moments, what are the next steps for you? You can follow the link that Arash has supplied in the chat, or that is currently on the slide, and register. By registering for our career space, you will receive reminders and additional information, both for this call and for future upcoming calls. This is still online, so you'll complete a profile which may not be specific to this call, so we sort of know who you are, and provide a current CV. And then for the call itself, please submit a cover letter and a project proposal that would explain in a few pages how you would address the challenge. The challenge itself will be laid out for you in a few moments, so please do pay attention. Please also read the call text that is on the website. Very important that your project proposal stays in scope, that you take a few notes and understand what the ask is, because out of scope proposals tend to get ranked lower, even if your CV is excellent. If you don't have the solution in your project proposal, that's fine. That's what the bootcamp is for. We are looking for proposals and individuals with potential and with enthusiasm. And if we can see those things in your application submitted by the deadline, which is October 11th, then the evaluation committee is likely to rank your proposal and your application quite high. And we will choose the 5 group leader candidates and 10 postdoc fellow candidates or research assistant candidates in the case that you may be looking to do a PhD with the team. Please also apply. And we will invite you most likely the 1st or 2nd week of November to join us 1 month later for the bootcamp from December 8th to 12th. And I can already anticipate that our social activity might be a visit to the Heidelberg Christmas market, which is really a fantastic opportunity, although that would have to be in the evening rather than during the morning. I'm just spitballing here, but let's think about that. And looking forward to seeing 15 of you there potentially. And for those of you who choose not to apply for this call or are not selected for the bootcamp, please join our, let's say, expanded family via our career space. You can follow the link here and you'll receive regular updates from us anytime a call is announced in another topic. And with that, I will close and we will pass it back to Arash.
Thank you very much. Now I would like to ask Kelly to please share her slides. Yes, please go on.
Hello, sorry, I was muted. Okay, so hi again, I'm Kelly Vandera, and Stephen is going to start this presentation first, outline who is Novo Nordisk, and then I'll take over to describe you the project proposal. So, Stephen.
Perfect. Thank you so much, Kelly. And you can move to the next slide. So as Kelly just mentioned, we'll start this part of the webinar with introducing to you Novo Nordisk and double-clicking a little bit on what we're doing within our research and development organization, and also highlight how and in what way we're driving innovation and the importance of collaboration with external partners to realize our goals and aspirations. And then after that, I'll hand it over to Kelly, where she will elaborate further on the details of the specifics of this project proposal. So next slide. So Novo Nordisk is a company with a more than 100-year heritage, having been established in 1923. And we're truly a global healthcare company in terms of our global footprint. Our research and development organization is home to many thousands of scientists, including myself, Kelly, and Marianne. And we work together, focused on advancing and bringing forward a single pipeline of drugs for the treatment of various serious chronic diseases. As an organization, research and development, we essentially cover a significant proportion of the value chain in a pharmaceutical company. So that is right from the beginning in terms of the idea exploration and conception of ideas, to maturing that idea into some data and proof of concept, and later to validating that proof of concept in various preclinical testing setups. Subsequently, we then collaborate closely with our CMC colleagues to facilitate that lead or development candidate to be scaled up, and then allowing us to actually test it in a clinical setting. And all going well, that ends with, or rather starts for our patients with a new product reaching the market. In terms of research and development, Novo Nordisk invests substantial amounts in progressing our ideas, concepts from research to development, and that's reflected that in 2025 we have invested more than 20 billion Danish kroner, which corresponds to more than $3 billion US. We are a really diverse organization, and that's in particular highlighted by the breadth of nationalities that comprise the research and development organization, with more than 70 different nationalities. Next slide. As a company, we have a breadth of therapeutic areas which we investigate and develop new treatments for. Reflecting our heritage in insulin, diabetes is a core therapy area for our organization, and additionally also other cardiometabolic indications, including obesity and the broader cardiovascular disease space. And moreover, we have also engaged in adjacent disease states such as chronic kidney disease, MASH, and also emerging therapy areas such as Parkinson's, Alzheimer's. We also invest heavily within rare diseases, and here it is principally those related to endocrine disorders or blood diseases such as hemophilia. And it's of course a collaboration in terms of developing new drugs, both combining the core competencies that exist at Novo Nordisk within these biologies and therapeutic areas with that of the technological competencies that exist in the research and development organization, be it the ability to design and develop new molecules, or as is the case for this proposal, formulate them or incorporate them into a drug delivery system. Next slide. As mentioned, we are a global healthcare company, and that's very much reflected within our research and development organization, which is located across 5 different countries. And across those different countries, the locations are also dictated in respect of what kind of activities, roles and responsibilities, which are anchored in different locations, which you can see in this map is reflected in different colorings. Our key global research and development hubs are principally located in the greater Copenhagen area, where myself, Kelly, and Marianne are situated, and also at our greater Boston site in the US. And then in addition to that, we have some more specific capability units, be it focusing on CMC, data science, or medical device research, which are located in our London, Seattle, and Fremont sites. And then beyond that, we also have specific discovery units that are more focused on very early stage target discovery. And in these settings, we have sites both in Beijing, China, Oxford, UK, and also in the US. Next slide. So as a pharmaceutical company, we value and put huge priority in how and what we innovate in particular with a focus on developing transformative treatment solutions to serve unmet needs as it relates to the chronic diseases which we seek to treat and in some cases cure. That is reflected in the number of patients which our drug products are currently treating. Today, approximately 45 million patients are treated for diabetes or obesity with innovative drug products developed at Novo Nordisk. And of course, this is something which we continue to drive in terms of bringing new innovations to the patient. A very good example of the transformative impact of innovations emerging from research and development at Novo Nordisk is the GLP-1 analog semaglutide, which has developed through research and clinical investigations, data supporting a broad range of benefits that go beyond the initial utility for glycemic control and weight loss, but also reveals positive impacts in terms of cardiovascular protection and also kidney and benefits. So of course, as a scientific and curious organization, research and development are at the heart of driving innovation for Novo Nordisk and looking to develop the next generation of treatments for patients living with serious chronic diseases. Next slide. And it goes without saying that innovation is a team sport. And for us, that also requires partnering and engaging with external partners to drive co-creation and co-innovation in a means such as that which we're engaging with BioMed X. So we recognize the importance of opening up key unmet needs and scientific questions which we maybe alone cannot solve, but which we recognize has the potential to be solved by working collaboratively with clever and curious minds that exist externally in the broader scientific community. And it is very much with that mindset that we have engaged with BioMed X and are stepping into this call proposal. And with that, Kelly, I'll hand it over to you to detail out more on the science behind the proposal that we're bringing forward in today's webinar.
Yes, thank you, Stephen. So yeah, let's jump into the scientific part. And let's begin by considering the shift from injectable to oral peptide drug delivery. Oral administration is non-invasive, is preferred by patients, and there's no need for healthcare professionals to assist with administration. In addition to that, it offers ambient temperature storage, which simplifies logistics and improves accessibility. So all these advantages make oral delivery a compelling alternative to injection. So despite its promise, oral peptide drug delivery still remains a challenge. And that can be seen by the fact that there are more than 10 technologies under development currently, yet only 2 have reached the market. So this gap highlights the need for innovation. So peptides as drugs face several biological and physicochemical barriers when it comes to their oral delivery. So they're prone to enzymatic degradation by pepsin in the stomach or trypsin, chymotrypsin, or other proteolytic enzymes in the intestines. Their high molecular weight and hydrophilicity leads to their low permeability and overall their low bioavailability. So all these parameters are, all these factors combined with the biological barriers make oral delivery particularly complex. I'm going to go through the biological barriers in the next slide. So the gastrointestinal tract offers a lot of physiological barriers, namely the residency time, motility, the complicated mucus layer, pH gradient going from acidic pH in the stomach to a more basic in the large intestine, and then of course fluids, in the presence of fluids. All these parameters are influenced by food intake, which adds another layer of complexity to the formulation design. So based on our experience, to overcome all those challenges, formulations need to achieve high local concentration of both peptide and permeation enhancer, minimize the effect of dilution by food intake or fluids, and ensure a prolonged retention of the dosage form and continuous release. In the following slides, I'm going to just go into each one of those factors into a little bit more detail, and I'm going to start with a high local concentration. We have shown that semaglutide and its permeation enhancer, in this case SNAC, concentrate to areas close to the tablet. Using endoscopic aspiration and imaging techniques, just a second to put my laser pointer. So you can also see here on the top 2 graphs that semaglutide and SNAC concentrate underneath the tablet. It is important here to mention that both semaglutide and SNAC concentrations are 10 times higher under the tablet compared to just even 6 centimeters away from it. So to have a more visual outcome of this observation, we pursued immunofluorescent and electron microscopy techniques to confirm this outcome. So in this presentation, I have included the immunofluorescent images that illustrate the localization of semaglutide predominantly to the surface mucus pit regions of the gastric epithelium. So in the top left image number A, if you can see, then you can see the immunofluorescent reactivity for semaglutide under the tablet, and this is shown in red. If you zoom in this picture, then you'll end up in the image 5B, and then you can see that semaglutide localization was mostly restricted to the surface mucous epithelial cells in the pit and neck region of the gastric mucosa. The 2 middle images, C and D, D is the zoomed in of this rectangular here in image C. You can see the most abundant signal for semaglutide in the deep intact layers where robust expression of tight junction protein was detected in the apical region, in the apical surface. So again, here in red, you see the signal for semaglutide, and in green, you see the signal from the tight junction protein. And then finally, semaglutide was also detected within the blood vessel. You can see that in the bottom right image, image F. 5 minutes after dosing. So overall, these findings underscore the importance of maintaining high local concentration of both semaglutide and SNAC at the epithelial surface for sufficient duration. Let's move on to the food effect. So food intake significantly impairs absorption. Here I've included two examples, one the oral semaglutide and the other one the oral calcitonin. Postprandial administration of oral semaglutide to healthy individuals nearly abolished semaglutide absorption. This is very clear by the two graphs included in the blue box, where they present the semaglutide concentration kinetic profile of the healthy volunteers in the fed and fasted state, respectively. You can see the detrimental effect of food is very obvious. Similarly, oral calcitonin shows reduced bioavailability with increased water and food volume. So in the pink box on your left-hand side, you can see a graph that presents the food effect of oral calcitonin. Here you can see a reduction as much as 74% from having a no meal to having a meal. The top, the black full circles present the no meal curve, while the rest are different time points of taking food along with the oral administration of calcitonin. In addition, when oral calcitonin was dosed with 50 milliliters of water compared to 200 milliliters of water, there was a twofold increase in the maximum concentration. And then you can also see that clearly. The top line is with 50 milliliters of water compared to the second line, which is the 200 milliliters of water. These findings illustrate how dilution effect can diminish the concentration gradient and therefore reduce the efficacy. Finally, prolonged retention and continuous release. Rapid gastrointestinal transit is already known that affects oral peptide formulation absorption. So we conducted a proof of concept study by repeating dosing of liquid formulations with constant SNAC concentration every 20 minutes for 3 times. This study demonstrated the potential of sustained peptide absorption, as you can see on the graphs on your right-hand side. The top graph is 2 doses of multi-dosing of those liquid formulations, and the second graph is the comparison of those 2 liquid formulations compared to the single dosing of the liquid formulation and the tablet administration. You can see here that the repeated dosing has an increased semaglutide concentration in the plasma. Therefore, ensuring a long retention time of peptides is a key factor for a successful gastric delivery of semaglutide. Our proposal is to develop an innovative oral formulation technology that can prolong the residency of the formulation in the gastrointestinal tract. The aim is to secure the proximity of the formulation towards the epithelium for at least 20 minutes, minimize food and water effect, and of course, be safe and scalable. Our aim is to enhance the passive diffusion without affecting the motility or the mucosal health. Open innovation just gives us opportunities to reach diverse expertise, researchers from all around the world with their fresh perspectives on the topic and field. And then this innovation approach that BioMed X is offering is also enhancing problem solving and therefore improving and finding more effective solutions. So we invite you to apply to this project proposal. And the candidates can have a background in pharmacy, biomedical engineering, chemistry, material science, but we are not limited to this. So any relevant background is also, we are also open to that. Candidates must hold a master's or a PhD degree and should have a strong interest in oral delivery of large molecules. A good understanding of the gastrointestinal tract is necessary. And what is really important for us is to be curious and open to challenges. So if you take any of the boxes here, any of the ticks here, then please apply. The winning team will closely be in collaboration with Novo Nordisk. They will gain access to scientific expertise by having regular meetings, and they will have access also to innovative research ideas and all the challenges of this exciting topic. During the process, formulations will be developed and a lot of formulations will be screened. If there is a very promising formulation and we have a lead composition that we are interested to test, then our animal models will be accessible to you to test in preclinical models and see the effective outcome of those formulations. And with that, I would like to thank you for your attention.
Thank you very much, Kelly. Thank you for this beautiful presentation. And with that, we're going to get to the Q&A.